So the following question has to do with additive genes, but this time we have a lot of different alleles that can affect it, right? And so the question goes that we have pigmentation in imaginary river bottom dweller mucus eucus, and it's a quantitative character controlled by a set of 5 independently segregating polygenes with 2 alleles each, A, B, C, D, and E, right? A pigment is deposited at 3 different levels depending on the threshold of gene products produced by the capital letters. So if you have a capital letter, it basically changes the threshold of the additive gene, right? So grayish brown pigmentation is seen if at least 4 capital letters or alleles are present, right? So if you have 4, 5, 6, 7, 8, 9, 10, then you have the grayish brown pigmentation. Light tan is if you have 2 or 3 capital letters, and then whitish blue is seen if those thresholds are not met. So if you have 0, 1, or 2. Now, if a true breeding homozygous dominant for all the gene animal is crossed with a homozygous recessive um, animal that's for all the genes essentially, and the progeny are intercrossed, what kind of phenotypes are expected in the F1 and the F2? Now, in the F1, it shouldn't be too hard. In the F1, you're going to have a pure heterozygote, right, at all of them. And so what you're going to end up having is 5 capital letters, right? You have 5 capital letters, and so we're going to see a grayish brown pigmentation. However, when we cross the F1 again with itself, we're going to an F2 generation, and since it's heterozygote at all the genes, we have basically 10 different positions, right? 10 different allelic positions in which we can organize capital letters and lowercase letters. And so the probability of having all capital letters, right, is essentially 1/2 multiplied 10 times through, right? Because at each position you have a 1/2 chance of having a capital letter for the F2 generation. And so it's 1 out of 1024 for basically the homozygous recessive or homozygous dominant at all the alleles. But we want to know like stuff like how many, like 2 capital letters, 3 capital letters, 4 capital letters, essentially like that. And one way to do this is, oh, looky here, the binomial expansion, right? If I use Pascal's triangle and keep expanding it all the way to the 10th row, we get this expansion right here. And this tells us a probability of 10 capital letters, 9, 8, 7, 6, 5, 4, 3, 2, and 1, and 0 capital letters, right? And this is in any order possible, right? So this is saying basically there's 10 ways of organizing 9 capital letters on these 10 allelic positions. Now, one way to do this is just look at essentially, oh, if we want to see a white pigmentation, we have to have less than 2 capital letters. So essentially, we have the probability of 0 capital letters and 1 capital letter, which is 11 out of 1024. These numbers add up to 1024 essentially. So that's for our whitish. Now, for our light tan, we have 2 or 3 capital letters, and 2 or 3 is 45 and 120 add up together, which is 165 over 1024. And so this is for our light tan. And then for 4 capital letters, or essentially our grayish brown, it's this and up, added up together, right? So I'm going to use a different color for that. We have 1 plus 10 plus 45 all the way, all these add up. And so what we get is 848 over 1024. And yes, I did that in my head and not by looking at the book. Now, another way we can actually find individual numbers instead of just doing Pascal's triangle, which is not mandatory, but you can do it, is using this equation right here, right? This is a probability equation where we have the dominant allele and the recessive allele, right? And then we have the total number of trials, which is n, factorial. And factorial is basically you multiply each number going down. So say if number was 5, then it'd be 5 times 4 times 3 all the way down, right? So we have n factorial over the number of dominant alleles that we have, or the capital letters in this situation, factorial, times the number of recessive alleles that we have, or the lowercase letters in this case, right? And this will be multiplied by the probability that you have a dominant allele, which is 1/2, to the power of the number of dominant alleles that you have. And so this would be, for example, D. And then for the probability of having a recessive allele, or the lowercase letter, is also 1/2 times to the power of the number of recessive alleles. And so this is kind of complicated, and that's why most people won't use it. And Pascal's triangle is actually a lot easier for binomial expansion. But say, for example, we had 20 different allelic positions, and we wanted to find the probability that you had 7 dominant alleles and 13 recessive. Then it would be kind of ridiculous to do the Pascal's triangle. You'd be expanding it forever. But if you just use this equation, we have the total number of trials, right? The total number of dominant alleles, recessive alleles, the probability of the dominant allele to the power of dominant alleles, and the probability of recessive allele to the power of recessive alleles, you can find the probability of having that specific genotype. And so this is how you do additive genes and the probabilities of their genotypes when they're pretty large.